nuclear chain fibers. So both of these have a purpose in as far as preventing our muscle from overexerting itself. But bag fibers, so what happens is we have a neuron that comes from the spinal cord, and it's actually going to synapse on a bag fiber, and the bag fiber, and then in turn, will have a neuron that comes and wraps around like this. Oops, now let's get the idea. And then that will actually go through and synapse here. So let me define what we're looking at first. So two things. The bag fiber itself, it's going to, both of these combined tell us about the intensity of a stretch. So we call this part our stretch reflex, meaning it's to prevent our muscles from overstretching. So with our bag fibers, what we see is it tells us about the intensity of our stretch. So how far apart are our muscles being stretched? And then it also tells us, and then our nuclear chain fiber, it tells us about the duration of the stretch. You know, we don't want to overexert a muscle for too long, so that's where, well, in part, where the nuclear chain fibers come in. So once we have this neuron come from the spinal cord, and then this neuron right here is called a gamma dynamic neuron. So that synapse is here, and that's the afferent connection from the spinal cord. Connects here, and that goes around the nuclear bag fiber, and as the muscle stretches and gets contracted, it feels the intensity of it. It will then signal to inside a cell body to a neuron in here. And we'll actually see a broken off pathway as well. These are both going to be EPSPs, but they're both going to connect to a different muscle, if they're going to have a different effector. And I'm going to write stuff over here, but it's important to understand that we're actually talking about the muscle which this bag fiber is on. But in order for this picture to make sense, I'm going to separate them apart. So really, this bag fiber is part of the muscle fiber that's giving feedback and thus ultimately affecting the muscle fiber itself. So we're going to have the agonist muscle. We're going to have the antagonist muscle. In the case of this route, the agonist and the antagonist. So let's say I need to go from here down to here. I have to tell my tricep to elongate, and I have to tell my bicep to, or other way around. Wait, no, that's right. Sorry. So I have to tell my tricep to contract. I have to tell my bicep to elongate. That's done through this system here, as well as if I was, doing something with my legs, like if I was standing up or sitting down, I'd have a relationship between my hamstrings and my quads. This signal right here is also, or this neuron is called type A1A neuron because it's very fast. You know, when you've had a lot of exertion and I need to move my arms, it's okay to instantly know, like, hey, I need to move my neurons now, or I need to move my arms now. We also see a connection from the spinal cord onto our nuclear chain fiber. And it's going to follow a very similar route to what our bag fibers did. Ultimately either activating or deactivating or activating our agonist depending on what scenario is going on. These are a little bit slower. We call these our class 2 neurons in order to make sure, or I guess where the difference comes in is 1A versus 2. 1A is a lot faster and 2 is a lot slower. So meaning if I needed to hold my arm out like this forever, the 2 neuron, or the nuclear chain fiber is eventually going to tell me like, hey, your arm's tired or you're overstretching. Whatever is going on with that neuron, it's going to be communicating that, so on and so forth. Then we have another structure built into our joints, or our tendons. Our tendons specifically. So as you can see here, I have our bone, and then I have a muscle, and in between we have a tendon, which connects bone to muscle. Between the muscle and the tendon, we have something called a Golgi tendon organ. You see, if our muscles were left to our own devices, if I wanted to lift 1,000 pounds, I could, if I had enough muscle. But in the process, I might also injure that connection to bone, or I may even hurt the bone itself if I provide enough force. So we have a small thing called the Golgi tendon organ, and that feeds into our system over here, and that's going to synapse differently. And that as well is going to feed to, let's say, our agonist. And that's going to prevent our agonist from actually contracting further. So it's actually going to send IPSPs to the agonist saying, hey, you need to stop doing whatever it is you're doing. You're going to hurt this bone. So if you're ever at the gym and you're trying to do a huge squat more than you're supposed to lift, you can blame your Golgi bodies for stopping you from doing that. But also, you should be telling them thank you. So all of this together makes up the stretch reflex, but then we also have something called the withdrawal reflex. So the withdrawal reflex occurs when, let's say, I touch something hot, and when I touch something hot, assuming there's a hot plate right here, I'm going to naturally withdraw, and then my body's actually going to force my other arm out and my foot back to get away from the pain. So in that moment, I'm having a cascade of cascaded signals go through or up from the sensory organ, which is my hand in this case, up through into my spinal cord, where the decision made is that, hey, activate this reflex system, back up, push away, don't like it, get away from me. And that's the withdrawal. All right, now we're going to talk about the old sniffer, not to be too on the nose about it. But when we breathe in air and there's something stinky around, so I don't know, let's say, here's that piece of chocolate that was in Brother Shaw's office the other day. Chocolate, right? Now, chocolate radiates smell, and that's going to go through the nose, and it's going to hit this whole deal here. So these are neurons that are connected to an olfactory bulb up here, but right here is our cribriform plate. This is our olfactory bulb. And then these would make those olfactory neurons. So once the smell hits these olfactory neurons, here's what's going to happen exactly. So right here we have adrenal cyclase. So adrenal cyclase is held in conjunction with like a mucusy smell, or a mucus, which then captures the smelly stuff or chemicals or particles or whatever it is. And that in turn, once the smell gets to there, remember that is really just this right here. But once it gets into the adrenal cyclase, it's going to release some level of CAMP. And then CAMP is going to, in turn, come and open up this channel up here. And then that is going to allow depolarization to occur, because this is just a cation channel. Oops. So we're going to let in calcium and sodium into our channels. That causes depolarization, and in order to balance out the charge, we have Cl minus, and it is going to leave the cell in conjunction. The level of depolarization allows us to smell different things, and that goes, and we have smell. All right, now we're going to talk about taste. So here we got our friend Licky Ricky. Now this is the guy in elementary that seemed to lick absolutely everything, and we're going to talk about why he liked to lick absolutely everything. So Licky Ricky has a tongue, hence the name. Licky Ricky has this thing. So what we're looking at is the surface of the tongue here. Now there's a couple of different structure names for that bump right there. But in general, we call them lingual papillae. It's a hard word. They come in varying shapes and sizes, but the point being is they're this mushroom-like thing that rises above the tongue. They provide a whole bunch of different functions. But off of that, we have actually our taste buds, or taste cells. Call this taste cell. So over here on Licky Ricky's taste cell, we are going to break down what each one of these look like. Now, every one of these taste cells actually is specified to one function, but for our purposes, I've generalized it to a single cell, so we don't have to look at a bunch of different pictures. So on one type of taste cell, we're sour and sweet. So with salty, what we are experiencing is what you would think, Na plus. So Na plus is going to come and it's actually going to go through this channel called an Enoch. And then when we're experiencing sour, we're also going to have hydrogen ions that come through this Enoch. When we're experiencing sweet, which is anything like sugars, so let's say we have a glucose or glucose-like thing, that's actually not going to go through the cell, but it's going to interact with the G protein receptor called phospholipase C. Point being is, if this is IP3, every single one of these in their own respect, if you're an ion, you're going to do it through BONG. If you're IP3, you're going to do it through ligand-gated mediation. But you're going to open up the SR to release its calcium. So calcium comes out, and it's going to interact with the vesicles and the snare proteins, twist, and the receptors are going to come out and interact with the gustal neuron. All right, now let's talk about hearing. So hearing is kind of complicated, but we're going to do our best, take our best shot at it. So the sound comes in through the ear, like your mother-in-law, just nag, 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 nag. Just kidding, we love our family. So that's going to go through, and that is going to vibrate these oscillatory bones right here. And the point with these bones is that when, let's say you have markers are actually a good example. Let's say as I wiggle this, notice how as I pivot on this end moves a lot more. Well, that's exactly what's happening from here to here, and here to there, and there to there. So by the time we get this, we have what's called a tympanic membrane right here. By the time that's vibrated, it's just gone, or by the time this vibrates the small one, the signal is just amplified however many fold until we get to the stapes, which is actually like a piston that goes in and out. So the stapes is acting like a piston on the fluid inside our cochlea, which is this snail-looking thing. Now, it's not an accurate drawing, but we work with what we got in a two-dimensional world. As it's pushing fluid inside the cochlea, which another name for that fluid is actually called perilymph. So as the perilymph is oscillated, eventually it's going to hit a point along this track here where it resonates with this red line in the middle. Now this red line in the middle actually represents another cavity, and what separates this perilymph from this cavity is something called the basal membrane. So let's say it's a low frequency and it's going to resonate up here, and it's going to cause the basal membrane to vibrate at that point. So coming to our cross-section picture here, there's a lot going on here, but we're going to describe it all. So this is our perilymph. And then that causes this, our basal membrane, which is this red line, to oscillate back and forth. And that's in turn going to cause our endolymph. Get it? 'Cause it's inside. Pretty cool. That's going to cause this tectorial plate to move, or tectorial membrane, whatever you prefer. As the tectorial membrane moves, what's going to happen is, you see these structures here, these are called hair cells. Now for the time being, we're actually going to focus on this hair cell in particular. When this hair cell moves, on the end of it, attached to the tectorial membrane, are actually tiny little hairs that we call stereocilia. The stereocilia, when this vibrates, it gets pulled or leans one way, and that actually causes a depolarization event for calcium to come from this structure over here, which is called the stria vascularis. And it is a vascularized component, and that's where we're getting the calcium from. That's actually going to flood into our endolymph here, and therefore into our hair cells, and cause a depolarization event in total. Talking about the stria vascularis, because typically when we talk about depolarization, we're actually talking about sodium entering a cell. But for our poor hair cells, that's actually not the case. We're going to be using potassium. So how the stria vascularis, that's this section in here, and then this is going to be our endolymph. inside the membranes of the stria vascularis, what we see is first a sodium and potassium pump. So we're going to kick out sodium, and we're going to bring in potassium, just like we'd see with a normal ATPase pump. Down here we have a different pump called an NKCC pump, type 1. So the NKCC pump, it's actually a secondary active transport, meaning it's relying upon the concentration gradient established when we pump sodium out, and that is going to bring in actually 4 ions in total, but 3 unique types. We're going to bring in sodium, we're going to bring in potassium, and we're going to bring in 2 chloride anions. Now when it gets to here, what happens is we actually have potassium leak channels. So potassium is going to leak out. In turn, on the other side of the stria vascularis, what we see is the exact same thing. So again, we're kicking out sodium, we're bringing in potassium. This potassium that we just pumped here, we're going to bring it in, we're going to bring our sodium in, we're going to bring our chloride ions in, and again, we have another chloride leak channel, and that's going to pump it into the endolymph. What we see eventually is that the endolymph has a really high concentration of potassium. So let's say it's like 150 millimoles, I think is the measurement that we have for that. So super high concentration, remember, high concentration here, low concentration of potassium here in the perilymph. It's 150, that's like 5. Inside our hair cells, we see a concentration of also about 150 millimole of potassium specifically. This is where it gets a bit weird because you look at it, it's like, well, how in the world is potassium going to cause a depolarization event of a hair cell? Well, to show that accurately, I'm going to need to erase our original ear diagram. Just kidding, you can still hear me, even though I erased the ear. You're still good. Don't freak out. Draw a box, and with my box I'm going to have my hair cell coming in like I showed you, and then I need, one sec, I have to reference my picture because I have to draw the right way. Hair cell. Right. Okay, so make sure I have this done right. So this is going to be my perilymph. Nope, other way around. This is my endolymph. This is my perilymph, and then down here, I'm going to make this larger. This is my hair cell. So we do see a concentration gradient established where, let's do this part blue. We see the inside of our cell become negative, and the outside of our cells becoming positive here. But between the endolymph and the perilymph is the whole white space, we actually see another concentration gradient established. Since we're so high, our potassium millimolar values, since we're so high in potassium on the outside here, we also see an additional concentration gradient established between here. So from a concentration gradient standpoint, we don't actually see any reason for potassium to want to go inside the hair cell. But as it turns out, from a concentration gradient or from an electrogradient standpoint, it makes total sense that potassium, if it has some leak channels on this hair cell, let's bust open some channels. Potassium is going to flow from here through the hair cell and out to the perilymph, where it will in turn be recycled back through the stria vascularis. That process ultimately causes a depolarization event inside our hair cell. Extend that again, where we have our SR, and again we have calcium comes out as the potassium ions come in and go out. That's because of their bond. And our vesicles are going to be released. That goes to the brain. So our vesicles get released, and then that goes out, and all of a sudden we're hearing things. Magical.